Hi, I'm Clariza ACEBES, and the video is about my learnings and realizations I got throughout this course.
Immunology began as a branch of microbiology. It is defined as the study of molecules, cells, organs, and systems responsible for the recognition and disposal of non-self material. How body components respond and interact, desirable or undesirable consequences of immune interactions, and how the immune system can be manipulated to protect against or to treat a disease. Considered as the father of immunology is Louis Pasteur. Host-parasite relation. The successful host-parasite relationship is the body's own microbial flora, and the most unsuccessful host-parasite relationship results to infectious disease. The host is a cell or organism which harbors another organism or biological entity, usually a parasite. This has a mechanism which the host defends itself against invasions of organisms. Non-specific defense mechanism and the specific defense mechanism, and under the specific defense mechanism is the humoral immune response and cellular immune response. Immunocompromised hosts are patients that could be very young and very old that does not have the ability to respond normally to an infection because of an impaired or weakened immune system. This is due to the pressure of the host defense mechanisms, non-specific, specific, or both, thus are prone to any opportunistic infections. Next, the parasite is an organism that lives on or in a host organism and gets its food from or at the expense of its host. Our body has a natural defense armor called the immune system. Our immune system, this gives us the ability to resist a disease or a particular infection, which is called immunity. Immunity has different types: the specific or adaptive immunity, the non-specific or natural or innate immunity. Non-specific immunity is also called as natural or innate immunity, which functions as the body's first line of defense against any infectious agents. Under non-specific immunity is the external defense system, where it's composed of physical, chemical, biological barriers that prevent infectious agents from entering the body, and internal defense system, where it is composed of both cells and soluble factors such as white blood cells and antibodies. Furthermore, non-specific immunity can be classified as species immunity, racial immunity, and individual immunity. On the other hand, specific immunity or acquired or adaptive immunity is a defense system that protects the body against pathogenic organisms and other types of diseases, such as cancer. Immunization is a process whereby a person is made immune or resistant to infectious disease, typically by the administration of a vaccine or vaccination. Vaccine stimulates the body's own immune system to protect the person against a subsequent infection or disease by inoculating dead or controlled kind of virus or disease. The main difference between the innate and the adaptive immunity is the mechanism and receptors used for immune recognition. The immune system is a system of multiple biological structures and processes within an organism that protects the body against diseases. The first line of defense against infection is unbroken skin, mucosal membrane surfaces, and secretions. The function of the immune system is to recognize self from non-self and to defend the body against non-self. The immune system's role is to defend the body against infections, which is the defense system, recognize and respond to foreign antigens such as their surveillance system, and also defends the body against development of tumors or cancers. Cooperation is required for optimal function of the immune system. This cooperation cooperative interaction in both specific cellular elements, cell products, and non-lymphoid elements. Cells of the immune system consist of lymphocytes, specialized cells that capture and display microbial antigen, and effector cells that eliminate microbes. The immune system develops when the embryo rises from the gut-associated tissues. In a very young embryo, the lymphoid stem cells are first produced by the fetal liver and yolk sac. Specific or adaptive immune response is highly developed and a more specialized mechanism of immune response, where the response is directed against specific not recognized by the host as self. There are three characteristics of immune response. First is the specificity. It's highly selective and discriminatory immune response. Second is heterogeneity, where it means that a wide variety of cell types and products are produced to interact with diversity of responses. And lastly, memory, where a property of accelerated or augmented specific immune response upon subsequent exposure to similar immunogens. There are two mechanisms that mediate specific immune response. The humoral immune response and the cellular immune response, which are a defense mechanism that forms the second line of body defense. Humoral immune response, or also known as antibody-mediated immunity, are responses that governed by the thymus-independent B cells, and antibody-mediated immunity to infection can be acquired if the antibodies are formed by the host or received from another source. Cellular immune response, are also known as cell-mediated immunity, are governed by thymus-dependent cells. This consists of immune activities that differ from antibody-mediated immunity. Cell-mediated immunity is moderated by the link between T lymphocytes and phagocytic cells. Serology is a subdivision of immunology, the branch of laboratory medicine concerned with the investigation of blood serum with special reference to its antibody content. In vivo reaction, antigens may produce cytotoxic effect or may produce antibody to neutralize antigen, while in vitro antibody-antigen reactions can be specific or non-specific, where it identifies the antigen of microorganisms or antibody in fluids. Sensitivity is the ability of a clinical test to be positive in the presence of homologous antigen, while specificity is the ability of a clinical test to be negative in the presence of its homologous antigen. In the nature of antigen-antibody reaction, it has three concepts: the lock and key concept, the covalent bonds, and reversible. Antibodies can be distinguished differences in structure, isomeric forms, and in secondary and tertiary structure of an antigen. There are four factors affecting the measurement of antigen-antibody reactions: affinity, avidity, antigen-antibody ratio, and the physical form of the antigen. Cell-mediated immunity is more difficult to define or quantify than humoral immunity. The reaction of antibody with its antigen can be detected directly or indirectly in a number of ways. In vivo tests, delayed hypersensitivity is a manifestation of CMI, which can be defined as an increased reactivity to specific antigen mediated not by antibodies but by T cells. In vitro tests are designed to measure either the proliferation of T lymphocytes in response to antigen or the production of lymphokines. Materials necessary for basic serologic techniques are glassware, such as test tubes, glass slides, and pipettes. Then, constant temperature devices such as incubator and water bath, and rotating machines required for antibody-antigen reactions. In handling specimens, make sure to mind standard precautions and make sure that the specimens are labeled. There are types of specimens that are used, which are serum, urine, cerebrospinal fluid and other body fluids, and swabs of various body exudates discharge. Dilution is the act of making a weaker solution from a stronger one, expressed as one unit of the original solution to the total of number of the units of the final solution. Serial dilution means decreasing the volume of serum progressively by maintaining a constant volume of the fluid. While in the determination of endpoint and titer, after serially diluting the patient's serum, equal amount of the antigen is added to each dilution to observe an immunologic reaction. The last tube that shows visible immunologic reaction is known as the endpoint of the test. The reciprocal of the greatest reacting dilution of the serum is considered as the measure of the titer or the concentration of the antibody. Then, in quality assurance and quality control, it is important to perform such in order to improve the accuracy and reliability of the testing, wherein this ensures the clinician ordering the test that the testing method has been done in correct and proper way as much as possible. Immunologic competence develops in an orderly manner in the very young and gradually improves to reach a peak of responsiveness in about time of puberty. The decline in immune response is due to the thymic involution that is the most controlling factor in the involution of the peripheral lymphoid structure. Immunity based on thymus-derived lymphocytes wanes earlier and more completely than those based on B cells. Thymus alone or thymic hormone alone cannot completely forestall the waning of the immunologic vigor with age. The most impressive correction of the immunologic aging is the transplantation of the thymus and stem cell from the bone marrow. The synthesis of circulating antibody as a response to a wide variety of antigenic stimuli has been shown to be relatively deficient in the old age. In contrast, increased antibody production to endogenous antigen or anti-nuclear antibodies and other autoantibodies occurs with great frequency during senescence. Even the declining of antibody production with aging can, or in large part, be explained in the basis of deficiency in the T helper cell population. Immunity to bacteria. It is when the bacteria possess an extreme degree of liability to change owing to factors such as mutation, metabolic alteration, lysogenization, production of toxins, and capability for prolonged survival. Bacterial infections can be classified as acute or highly productive type, chronic type, and toxigenic or toxic producing type. Immunity to fungi. Fungi can be categorized into four kinds: superficial mycosis, subcutaneous mycosis, respiratory mycosis, and commensals. Immunity to viral diseases. Viruses are unobligate intracellular parasites. A complete viral particle of viron may be regarded as a basic block genetic material, consisting of either DNA or RNA and surrounded by protective coat of protein that may also serve as a vehicle for transmission from one host to another. Some viruses are surrounded by a viral envelope consisting of a bilayer membrane containing viral glycoproteins that produces as spikes. Graft or transplant. It is a transfer of living cells, tissues, and organs from one part of the body to another. Graft versus host disease or GVHD can be an intentional consequence of blood transfusion or transplantation in severely immunocompromised or immune-suppressed patients. There are types of rejects or rejections caused by transplantations, which are graft rejection, hyperacute reaction, accelerated rejection, acute rejection, and chronic rejection. For immunosuppression, immunosuppressive agents are used in several ways, including induction and maintenance of immune suppression and treatment of rejection. A combination of different agents are frequently used to prevent graft rejection. However, the immunosuppressed state, when graft survival induced by this patient, comes at a hefty price, where it increased susceptibility to infection, malignancies, and other associated toxic side effects. Oncology is the study and the treatment of tumors or neoplasm. Neoplasm is the proliferation of cells that produce a mass, benign or malignant. These arise from the mutation of stem cells. Benign is a slow-growing, usually encapsulated, non-spreading, minimal mitotic activity and resembles parent tissue, while malignant is invasive and disseminated capacity for self-renewal and to continue the expansion of the malignant cell population. Our body has tumor suppressor genes, also called as anti-oncogenes. This is where it regulates the proliferation of cell growth in normal cells, and when inactivated, a block to proliferation is removed. A beautiful part of our body's defense system is that we have a defense armor against cancer. However, if the growth of malignant tumors and malignant cells evade the host defense, the cancer cell will spread and metastasize, which would affect other parts of the body, thus spreading. Maternal-fetal relationship. Theories of immune avoidance. There are ways in which avoidance is accomplished. First is lack of antigenicity. Fetuses are immunogenic wherein as early embryo they have transplantation antigens. 2. The uterus is a privileged site since a transplant can grow without being destroyed by an immunologic reaction. Third, impairment of maternal immune response where infections are more frequent during pregnancy, suggesting an impairment of the effector cells of cellular immunity. Fourth, placenta as an immunologic barrier. It separates the maternal and fetal circulation, which acts as a barrier. And 5. Protective antibodies. Antibodies can depress cellular responses. This is called immunological enhancement in tumor-host relationship and blocking in most other situations. Immunologic tolerance. It is the acquisition of non-reactivity towards particular antigens. Failure to mount an immune response to an antigen. Immunologic tolerance can be natural or self or induced. Self-recognition or tolerance refers to the critical process and failure to recognize self-antigens, which can result in autoimmune diseases. There are two types of tolerance, the central and peripheral, and deficits in both central and peripheral tolerance can cause autoimmune diseases and can contribute to asthma, allergies, and inflammatory bowel diseases. There are three mechanisms of the tolerance to self-antigen. First is the central tolerance by clonal selection. Second is the peripheral tolerance by cell deletion and inactivation. And third is the suppressor T cells. T-cell tolerance is that T cells do not show a marked difference in tolerance at different stages of maturation. Autoimmunity. It is defined as the breakdown of the immune system's ability to discriminate between self and non-self. Autoimmune disorders are serious chronic illnesses where the body's own immune system attacks its own organs. There are factors that can influence the development of autoimmunity. First is the potential for autoimmunity, where lymphocytes that are potentially reactive to self-antigen that exists in the body, and antibody expression, which are influenced by genetic factors, patient's age, and exogenous factors like radiation, drugs, or viruses. Immunopathologic mechanisms. Autoimmune disorders prevented by normal functioning of the immunologic regulatory mechanisms. Any dysfunctions may lead to production of autoantibodies that bind to self-antigens in the circulation, forming immune complexes or antigens in tissue. Examples of autoimmune diseases and disorders are vasculitis, carditis, Hashimoto thyroiditis, Graves' disease, type 1 diabetes, rheumatoid arthritis, SLE or systematic lupus erythematosus, celiac disease, and many more. Hypersensitivity are normal but exaggerated or uncontrolled immune responses to an antigen that can produce inflammation. It can be classified as immediate, antibody-mediated, or delayed, cell-mediated based on the time after exposure to offending antigen. Immunologic reaction dependent on the host response to a subsequent exposure of antigen is called immunization or sensitization. Allergy refers to an altered reaction to external substances, and atopy refers to immediate hypersensitivity mediated by IgE antibodies. Allergen refers to antigens that trigger allergic responses or reaction for a lower molecular weight substances that enter the body through inhalation, ingestion, or administered drugs. These are the types of antigen that can cause allergic reaction, which are environmental substances, infectious agents, self-antigens, and food. Going all through these chapters gave me the realization of the importance of immunology and serology. The principles and practice of immunology and serology affect every aspect of the clinical laboratory. Immunology and serology have come to represent the bedrock of laboratory diagnostics by underlying principles or practical applications.
This has been Clariza ACEBES. Thanks for watching.